Thank you. Well, it really is an honour to be able to talk to you about my research here today, and I'd like to thank the Academy, A, for admitting me, and B, for inviting me to give a presentation. And what I'd like to talk to you about this evening is one aspect of the work that we conduct within the Institute of Medical and Biological Engineering at the University of Leeds. And that is acellular biological scaffolds for functional tissue repair and replacement. But I'll begin by attempting to put the work that we do into context. There are clinical needs for regenerative solutions to meet the needs of society today. We have an aging population with an increased life expectancy. And the elderly, and I include myself in that, expect a good quality of life with high levels of activity. However, as we grow older, our tissues fail. They fail due to illness or injury or simply the aging process. And this can lead to significant loss of quality of life, particularly in individuals with chronic diseases. Unfortunately, that degeneration can occur from early middle age. And the simple fact is that around 10% of the population will need a tissue repair or replacement at some point during their lifetime. There are, of course, current options for tissue repair and replacement, but all of these have limitations. And none of these fully restore joint function, tissue function, and the quality of life. And the majority of them fail, of course, to grow and develop with the individual. Regenerative medicine, a new discipline, offers a promise to revolutionize patient care in the 21st century. However, it's imperative to ensure that any new regenerative medical technology solutions for the aging population meet not only the clinical needs and increased patient expectations, but are also affordable in order that they are clinically adopted. If we think about the therapeutic options in regenerative medicine, then different researchers pursue the cells, usually some form of stem cell. Others pursue biologics, pharmacological agents that can be used to restore normal cell function. And others pursue complex biomaterial scaffolds and matrices. Indeed, the conventional view of tissue engineering holds that all three elements will be necessary. In other words, if we combine cells with a biomaterial scaffold, add some biological factors, and produce a living tissue in the laboratory, that can then be implanted into the recipient to restore tissue function. The approach that we've taken within IMBE at the University of Leeds is unique in the UK. And the approach that we've pursued is to develop acellular biocompatible biological scaffolds, which can be implanted and then harness the patient's own cells for self-regeneration. Acellular biological scaffolds can be derived from both human donor tissue or animal tissue. And from a regulatory perspective, they can be delivered as human tissue products or class 3 medical devices. So why is this important? It's important because approaches that use cells and biologics are regulated like medicines, which means they have to go through phase 1, phase 2, and phase 3 clinical trials. Whereas human tissue products and medical devices go through the equivalent of a small phase 3 clinical trial with a low number of patients, before either being approved by the HTA or gaining CE mark and being released onto the market. And this makes the time and the cost from taking an idea from the laboratory through to the bedside for patient benefit very much the cost lower and the time very much faster. So what is the concept of a functional biological scaffold? Well, I've tried to illustrate it here. We begin with the tissue that we wish to replace. So if we wish to replace a heart valve, we begin with a heart valve, a ligament, a ligament. And if we look at that tissue microscopically, what we find is it essentially has two elements: the cells, which are stained blue, and the extracellular matrix, or scaffold, which is stained pink. Now, it's the cells which are immunogenic. It's the cells which act as the barrier to the free transplantation of tissue from one individual to another and from animals to man. So if we can treat the tissue using a process to remove those cells to produce a biological scaffold, which only contains the scaffold or the matrix, and the matrix is relatively well conserved among higher species and has low immunogenicity. When that is then implanted, it will regenerate with the patient's own cells. Now, we began to investigate this idea around about 2000, and we began by developing a process using porcine heart valves as a model. We developed a process, the process is patented, and we licensed the process to NHS Blood and Transplant Tissue Services, such that they could apply the technology to human tissues for the benefit of patients within the NHS. We then went through a series of academic studies in which we characterized the matrix, we did the biology, the biomechanics, and all of this was very successful. We tested the biocompatibility in small animal models, and again, that was successful. So we were ready to do preclinical studies. And we set up a collaboration with a surgeon in Brazil called Francisco da Costa, where we undertook studies of the acellular porcine aortic valves in sheep in order to try to prove the concept. In this slide, I've just shown the valve before decellularization and after decellularization, showing that it retains its physical form. We've removed the cells and the major antigens, and importantly, when tested in hydrodynamic tests, the acellular valves still function, and they show normal leaflet kinematics. When implanted into the sheep, after 6 months, the acellular valves have become recolonized with the sheep cells. And those cells are of the correct phenotype. And this was in marked contrast to controls that we did of living allogeneic valves in the same model. So that study demonstrated the proof of concept. The cardiac surgeon that we worked with was so impressed with our technology that he asked us if we would train his technicians at the Heart Valve Bank in Curitiba, Brazil, in the technique, which we gladly did. They began decellularizing human donor cardiac valves, and he began implanting them in patients. He's now implanted over a couple of hundred acellular cardiac valves in patients, and the early to medium-term results of those valves, as you see here in the red, are looking extremely good. So this collaboration was pivotal in our subsequent success. Between the period of 2003 and 2006, we began to apply our technology to a range of different tissues, such as pericardium, blood vessels, tendon, meniscus, and skin to produce an acellular dermis for application in wound healing. This research generated an additional 3 patent families. And so, around about the beginning of 2007, the decision was made that the technology should be spun out as a company. And Tissue Regenics was produced in order to commercialize our intellectual property. We continued to work with NHS Blood and Transplant with human tissues, and indeed, NHS Blood and Transplant collaborate with now the AIM-listed company, Tissue Regenics. This has led to further products being used clinically. Tissue Regenics market the D-cell vascular patch, and acellular dermis, D-cell dermis or Dermapure, is used to treat chronic, non-healing leg and foot ulcers. An independent clinical trial of the D-cell dermis in patients who had previously recalcitrant wounds showed quite exceptional results, with an 87% reduction in the wound area over a 6-month period. In addition, Tissue Regenics have a further couple of products in clinical trial, and NHS Blood and Transplant are setting up GMP manufacturing processes for acellular allogeneic valves. So what are we doing now? Well, the challenge that we're now addressing is that of osteoarthritis, particularly in the knee joint. We've been developing acellular bone soft tissue composite grafts, which we hope will be used for the replacement of damaged joint tissues. I think most of you will be aware that osteoarthritis is an extremely debilitating disease. It affects around 20% of the older population. But importantly, osteoarthritis can set in when any of the soft tissues within the knee joint are damaged. So our idea is if we can develop acellular osteochondral, bone tendon bone, and bone meniscus bone replacements, we can intervene early in the disease process to delay or maybe even prevent the development of osteoarthritis. I'll just say a little bit more about the work we've done on bone tendon bone. This is a project we've been doing in collaboration with NHS Blood and Transplant. They currently supply bone patella tendon bone to surgeons for ACL replacement. However, because that contains cells, usually dead and dying cells, when they're implanted, they show delayed remodeling and failure to integrate early. And this fails to restore full joint function. And what we hypothesize is by removing the cells, which we've done here, that we will get faster regeneration and incorporation and restoration of joint function. Importantly, when we decellularize these bone tendon bones, they retain their strength. We've recently completed a proof of concept study, again using sheep, where we've implanted the acellular human bone tendon bone as an ACL replacement in the sheep knee. And after 6 months, we've shown really good results with regeneration of the tendon, with ligamentization, and osseointegration in the bone tunnels. and NHS Blood and Transplant are now beginning to set up a GMP manufacturing process in order to validate that and begin to supply these acellular grafts. So to summarize the current state of play with this research, this chart illustrates the breadth of our technology, which can essentially be applied to any connective tissue. It also illustrates where we are in the research and innovation pipeline, with those products that are now used clinically, those in clinical trial, those in development by our strategic partners, either NHSBT or Tissue Regenics, and those which are still at the university research phase. And finally, I'd like to say that the rewards of doing this type of research are not just the social and economic benefits. In the studies we've been carrying out with the cardiovascular tissues in sheep, when we study the regeneration of the tissues over time, we've been finding that a particular cell type, the macrophage, M2 macrophages, invade the tissue very early on in the process, and they seem to pioneer the regeneration and constructive remodeling of the acellular grafts. We're only just beginning to study this in any detail, but the tissues we have from our animal studies provide us with a very rich source of material where we can ask fundamental questions about the regeneration process, and hopefully that will keep me busy for many years to come. So I'd like to stop there and thank you for your attention and thank our funders and collaborators.